Chapter 20 is Genomics and other omics. Genomics attempts to understand the content, organization, function, evolution of genetic information on genome. It has an impact on knowledge, human health, and agriculture. To study genomics, usually scientists map a genome, and they can do this through two ways. Genetic map, also called linkage map, which shows the relative distance between two genes. It's based on rates of crossing over or recombination, and it's expressed in units of percentage of recombination. The genetic map is crucial for determining the physical map, and is also crucial for whole genome sequencing. There are approximately, because it studies every base pair in the entire genome, and it has been done for over 1,000 species. The physical map, however, is a direct analysis of the DNA sequences, the number of base pairs. It is of higher resolution, more accurate, shows every gene, restriction mapping, and DNA sequencing. Whole genome sequencing, as I mentioned, is for every base pair in every genome, and it has been done for a lot of species. And what can whole genome sequencing show us? It can show us the single nucleotide polymorphisms, which are the DNA variations in a single base pair. These single nucleotide polymorphisms are equivalent to different alleles. Members of the same species may share similar but not identical DNA. There is no phenotypic differences among species with single nucleotide polymorphisms, and there are valuable markers in linkage studies. Single nucleotide polymorphisms are present throughout all human populations, and there are approximately 1 SNP for every 1,000 bases. So, for example, we have that person 1 might have a C in this position, but another person might have just a T in the position of the C, or an A in the position of the G, and everything else remains the same. So millions of single nucleotide polymorphisms have been mapped to a human genome. So this is part of genome-wide association. Single nucleotide polymorphisms compared to individuals with specific diseases to analyze the linkage of these SNPs to specific diseases. For example, approximately 520,000 people analyzed for stroke served to identify 32 loci associated with stroke risk. Non-random association between SNPs indicate close ties to a disease, such in the stroke example I just mentioned, macular degeneration, which are 100,000 SNPs on chromosome 1 approximately, and homosexuality has also been found to be genetically influenced. Haplotypes are DNA variations on a single chromosome or part of a chromosome. They are also linked to SNPs and sometimes are inherited together. There is genetic variance, ID genetic variance in the population, and the HapMap project catalogs SNPs in 4 human populations. Copy number variations affect the phenotype and are related to certain kinds of diseases. They are, as the name says, variations in copy numbers of DNA sequences, and these can be done. This can be due to a mechanism of deletion, one copy, or duplication. And there have been approximately 800 genes identified to be involved in autism, and out of these, approximately 30% has been attributed to copy number variations. So genomes have human genomes has also been important in evolution. Evolution and genome studies in evolution study the genetic traits of today's people to the hominid ancestors. And one example like is the Neanderthal and the Cheddar Man, and the comparison between the DNA of this specimen and modern human DNA. And it has also been important to tell apart the differences between carnivores, herbivores, and omnivores from an evolutionary standpoint. Since carnivores, for example, have expanded gene families for muscle myosin complex, for example, use an actin cytoskeleton components used for chewing. And there has been contracted gene families for starch and glucose metabolism because their diets are mostly protein, and loss of glucokinase regulatory protein for the glucose that forms part of the glucose metabolism as well. And this has not been found as much in herbivores and omnivores, but a similar example happens in wolves and dogs. Since approximately 20 to 40,000 years ago, dogs were evolved from wolves. And basically, this is due to a mutation in the WBS-CR17 gene, which is associated with impaired thinking and hypersociability. And this gene has been found very pronounced in dogs, and also the GTF2I gene and the GTF2ID1 gene, since these genes are responsible for social behavior. Dogs the small dogs usually have ACC allele. The medium dogs have a CNT alleles, and the big dogs have two copies of a different T alleles. Lastly, we have metagenomics, which are large genomes of a specific population or habitat that is sampled and determined, and it's applied mostly to microbial communities, but it also can be applied to humans and the human microbiome, which is present in the human gut. And it's been found that the environment shapes a lot of the microbiome composition, and approximately only 8% of human microbiome is heritable. And this has association with body mass index and how the body metabolizes cholesterol, for example. And another application of metagenomics is functional genomics, which determines the biological function of genes through the study of the transcriptome, which are all the RNAs transcribed from a genome, or the study of the proteome, which are all the proteins encoded by the genome. And this allows the determination of the biological function of both of these. So the transcriptome, all the mRNA is extracted from the organism, as we mentioned previously. It's used to make complementary DNA with the reverse transcription, and then the transcriptome can be studied. To predict protein function, we can study three types of genes: homologous genes, which are genes that are evolutionary related; ortholog genes, which are homologous genes in different species that arise from a common ancestor; or paralogs, which are genes with our homologous genes within the same organism that are due to gene replication. Comparative genomics is also a field that studies the similarity of gene content, function, and organization among genomes of different organisms. For example, humans share approximately 95 to 99% resemblance with the genome of mice, 96% with chimpanzees, 50% with fruit flies, 18% with plants. Gene function being homologous is when genes of similar sequences are expected to have a similar function, and a gene in one species may be equivalent to another gene in another species. Proteomics, as we mentioned, is studying the proteins encoded by the genome, and this is analyzed. This is analyzed by extracting the proteins of the organism, and this is difficult, and it's more difficult than DNA study. And the proteins are submitted undergo the two-dimensional page, a two-dimensional page, in which what remains in the page undergoes mass spectrometry or X-ray crystallography or nuclear magnetic resonance in order to determine the protein structure.